Introduction to the cardiovascular system, the composition and anatomy of blood, and the functions of the components. Blood is a connective tissue made up of cellular elements and an extracellular matrix. The cellular elements are referred to as the formed elements and include red blood cells, white blood cells, and platelets. The extracellular matrix, called plasma, makes blood unique among connective tissues because it is fluid. This fluid, which is mostly water, perpetually suspends the formed elements and enables them to circulate throughout the body within the cardiovascular system. Blood makes up approximately 8% of adult body weight. In the laboratory, blood samples are often centrifuged in order to separate the components of blood from one another. Erythrocytes are the heaviest elements in blood and settle at the very bottom of the tube. Above the erythrocyte layer, we see the buffy coat, a pale, thin layer of leukocytes and thrombocytes, which together make up less than 1% of the sample of whole blood. Above the buffy coat is the blood plasma, normally a pale, straw-colored fluid, which makes up the remainder of the sample. In normal blood, about 45% of a sample is erythrocytes, which is referred to as the hematocrit. The hematocrit of any one sample can vary significantly. However, about 36 to 50% according to gender and other factors, not counting the buffy coat, which makes up less than 1% of blood. We can estimate the mean plasma percentage to be the percent of blood that is not erythrocytes, or approximately 55%. Blood plasma. Like other fluids in the body, plasma is composed primarily of water. In fact, it is about 92% water. Dissolved or suspended within this water is a mixture of substances, most of which are proteins. The major components of plasma and their functions are summarized. Hemopoiesis and hematopoiesis. The lifespan of the formed elements is very brief, although one type of leukocyte or memory cells can survive for years. Most erythrocytes, leukocytes, and platelets normally live only a few hours to a few weeks. Thus, the body must form new blood cells and platelets quickly and continuously, a process known as hemopoiesis. In children, hemopoiesis can occur in the medullary cavity of long bones. In adults, the process is largely restricted to the cranial and pelvic bones, the vertebrae, the sternum, and the proximal epiphysis of the femur and humerus. Throughout adulthood, the liver and spleen maintain their ability to generate the formed elements. This process is referred to as extramedullary hemopoiesis. When a disease such as bone cancer destroys the bone marrow, causing hemopoiesis to fail, extramedullary hemopoiesis may be initiated. All formed elements arise from stem cells of the red bone marrow called hemopoietic stem cell or hemocystoblast. Hemopoiesis begins when the hemopoietic stem cell is exposed to appropriate chemical stimuli, collectively called hemopoietic growth factors, which prompt it to divide and differentiate. One daughter cell remains a hemopoietic stem cell, allowing hemopoiesis to occur. The other daughter cell becomes either of two types of more specialized stem cells. Erythrocytes. The most abundant formed elements in blood, erythrocytes, are basically sacs packed with an oxygen-carrying compound called hemoglobin. Production of erythrocytes in the red bone marrow occurs at the staggering rate of more than 2 million cells per second. For this production to occur, raw materials including iron, copper, zinc, B vitamins, glucose, lipids, and amino acids must be present in adequate amounts. Erythrocytes live only 120 days on average and must be continually replaced. Worn out erythrocytes are phagocytized by macrophages, and their hemoglobin is broken down. The breakdown products are recycled or removed as waste. Leukocytes. Leukocytes protect the body against invading microorganisms and body cells with mutated DNA, and they clean up debris. Thus, they are a major component of the body's defenses against disease. Leukocytes routinely leave the bloodstream to perform their defensive functions in the body's tissues, where they are often given distinct names such as macrophage or microglia, depending on their function. They leave the capillaries, which are the smallest blood vessels or other small vessels, through a process known as emigration or diapedesis, in which they squeeze through adjacent cells in a blood vessel wall. Once they have exited the capillaries, some leukocytes will take up fixed positions in lymphatic tissue, bone marrow, the spleen, the thymus, or other organs. Others will move about through the tissue spaces, sometimes wandering freely and sometimes moving toward the direction in which they are drawn by chemical signals, a mechanism known as positive chemotaxis. Lymphocytes. Lymphocytes are one of the types of leukocytes and will be discussed since they tie into the next set of body's defenses. The three major groups of lymphocytes include natural killer cells, B cells, and T cells. Natural killer cells, or NK cells, are capable of recognizing cells that do not express self-proteins on their plasma membrane or that contain foreign or abnormal markers. These non-self cells include cancer cells, cells infected with a virus, and other cells with atypical surface proteins. B lymphocytes, or B cells, and T lymphocytes, or T cells. These cells play predominant roles in defending the body against specific pathogens, such as disease-causing microorganisms, and are involved in specific immunity. B cells undergo a maturation process in the bone marrow, whereas T cells undergo maturation in the thymus. This site of maturation process gives rise to the name B and T cells. Plasma cells, a type of B cell, produce the antibodies or immunoglobulins that bind to specific foreign or abnormal components of plasma membranes. T cells provide immunity by physically attacking foreign or diseased cells. Memory cells are a variety of both B and T cells that form after exposure to a pathogen and mount rapid responses upon subsequent exposures. Unlike other leukocytes, memory cells live for many years. Platelets. After entering the circulation, approximately 1/3 of the newly formed platelets migrate to the spleen for storage for later release in response to any rupture in a blood vessel. They then become activated to perform their primary function, which is to limit blood loss. Platelets remain only about 10 days, then are phagocytized by macrophages. Platelets are key players in hemostasis. the process by which the body seals a ruptured blood vessel and prevents further loss of blood. Although rupture of larger vessels usually requires medical intervention, hemostasis is quite effective in dealing with simple wounds. There are 3 steps to the process: vascular spasm, the formation of a platelet plug, the coagulation or of blood clotting. Failure of any of these steps will result in hemorrhage.